Welcome to students. You all now know that cells were first discovered by Robert Hooke in year 1665. He observed the cells in cork slice using a primitive microscope. It was Robert Brown who discovered the nucleus in the cell. Purkinje in 1839 coined the term protoplasm for the fluid substance present inside the cell. The cell theory that all plants and animals are composed of cells and cell is the basic unit of life was presented by two biologists, Skeleton and Schwann. The cell theory was further expanded by Virchow by suggesting that cells arise from pre-existing cells. With the discovery of the electron microscope, it was possible to understand the cell organelles inside the cell. Cells have different shapes and are of different sizes. In the pictures you see, you have the red blood cell, then the columnar epithelial cells, then you have the large ovum cell, then there is the smooth muscle cell, the bone cell, the nerve cell and the sperm cell. Red blood cells are oval and small to carry oxygen and move with great speed from the heart to different parts of the body, and carry back carbon dioxide from different cells to the heart, the lungs. The ovum is large and non-motile with a lot of fatty material for the developing zygote. The muscle cells are spindle shaped because of their capacity to stretch. The bone cells are solid and small in size. The nerve cells look like wires and cable because the function of theirs is to transmit messages from the brain to different cells and back the cells to the brain. The sperm cell is motile cell, so it looks like a tadpole with a movable tail at its back. Let us perform two experiments in this lesson to observe plant cell and animal cell. The first one is to prepare a temporary amount of onion peel and observe the plant cell. The onion bulb has to be taken, cut with a knife, and a very thin section of the inner membrane taken out, stained with saffron stain and observed under the microscope. The detailed explanation and activity will be attached with this particular lesson for you to observe as a video. Now, after the slide is prepared, the observations that you see are: the cells have got a honeycomb-like pattern with a distinct cell wall and cell membrane. There is a centrally placed nucleus, or sometimes the nucleus is towards the periphery of the cell wall. There is dense cytoplasm present inside the cell. This is what you can see under a compound microscope. A neat picture of the same has been drawn both in the classwork and the record notebook. The next section of this experiment is to observe the cheek epithelial cells for identifying and looking at the animal cell. The cheek epithelial cells are loose cells available on the inner lining of the cheek. They can be removed easily with a toothpick placed on a slide. A drop of methylene blue stain is added to it and covered with a cover glass and observed under the microscope. The cells are different from the onion peel cells. They are oval type cells with a dense cytoplasm and a centrally placed nucleus. Only cell membrane is present, no cell wall is present. The nucleus is present at the center and small vacuoles are present which are many in number.